Hello, everyone, and welcome. Today we have a presentation on antigen presentation. A lot of these concepts are going to be review, and then some are also going to be new. But in any case, they will come up again and again and again for further consolidation. I believe in you that you will get it. However long it takes, you will get it. If I can be a little bit part of helping you get it, then that's the whole point, right? So let's orient ourselves. Where are we? So recall that the whole point of having T cells is to have like these very nosy cells that can look for antigen um, inside of other cells, right? We established that a lot of the mechanisms in place to detect antigen are primarily extracellular. You have a couple intracellular ones here and there, like the toll-like, some specific toll-like receptors on the macrophages, but by and large, antigen is if it's hiding inside a cell, it has less probability of getting picked up. Therefore, we have T cells to help out with that, and they do this. They detect intracellular antigens hiding inside cells, through major histocompatibility complexes, or MHCs for short. And you're going to hear about MHCs a lot. And they even get their name because when you talk about graft rejection and compatibility for like an organ transplant and things of this nature, MHC and their associated genetics become quite important. But that's not really the topic of discussion today. But it exists, right? So again, the T cells don't stick up their arms into other cells and feel for antigen. No, they have to use these MHC molecules. And these MHC molecules are essentially a sample platter of the protein environment or the antigen environment inside a cell. So if there is an abnormal or pathogenic type antigen inside the cell, on this platter, sample platter of MHC, a sample of that antigen could be projected, and the T cell will know that there is something hiding in the cell. There's two types of MHC though, Mxc Class I, and this all nucleated cells have it, and you have Mxc Class II. These your professional antigen presenting cells have it, right? And so when I was an undergrad a long time ago, I once asked this question in class, and I got laughed at by the professor. I don't find it very funny, but in hindsight, I suppose it is funny. I don't know. But because MHC Class One is all nucleated cells, that does mean that your professional antigen presenting cells have MHC Class One in addition to MHC Class Two. So it's not like you have one or the other. But no. In the case of professional antigen presenting cells, they uniquely have MHC Class Two, but just like any other nucleated cell, they also have MHC Class One, right? Kind of intuitive, I think, but undergrad was a different time. So we talk about the genetics for MHC Class 1. There are three main genes you should know about: HLA-A, B, and C, and these are on chromosome 6. We spoke about chromosome 14 in a past lecture. We're talking about coding for the B cell receptor. This has nothing to do with that, just because chromosome might become like a ding word in immunology. There's multiple chromosomes attributed to different things, just as there is anything you talk about in genetics, right? And I do not claim to be an expert in the subject. Actually, I would say it's probably one of my weaker subjects. I have a lot of weak subjects, but that's aside the point. So MHC, in terms of diversity, there really isn't that much, but they do tend to be pretty a broad category, right? So just keep that in mind. And we're not going to talk about the specifics of what they go into, but just know you get 3 from each parent, and so you're going to have 6 MHC Class 1 genes, and in a sense, you have like 6 kind of subtypes of these MHC Class 1, and then they operate or help out with specific classes, if you will, of antigens. And one of these, like, say, could be like acidic antigen or have like a certain type of protein sequence on them, things of this nature, beyond the scope of these introductory type modules. But if we talk about structure, and we did this in an earlier lecture, let's just say you have a phospholipid bilayer, okay, on the cell, specifically any nucleated cell, right? Because it's MHC Class I, right? You're going to have the different components going on. So you're going to have the transmembrane component, and you're going to have alpha 3, and you're going to have alpha 2, and you're going to have alpha 1. And tethered off alpha 1, you're going to have something called the beta 2 microglobulin, right? And so this is very important to note that the beta 2 microglobulin is unique to MHC Class 1. Now, MHC Class 2 actually has a beta chain. Now you notice that MHC 1 does not. These here are alphas, right? We started with 3, and you had 2, and then you had 1, and then down here is the beta 2 microglobulin. MXC Class 2 has the beta chain, and the MXC Class 1 does not. So to kind of substitute for its lack of beta, MHC Class 1 has a beta 2 microglobulin, which is like a knockoff. A loss of better words. Not really knockoff, I'm sure it's important, but that's structurally the difference, right? And again, HLA A, B, and C on chromosome 6 is going to be the genetic component on typically what you should know for this, right? Now, we spoke about this in the previous module. MXC1 also have this is like an alternate view. So this is like the same thing, but it's like alternate view. They have a groove. So when an antigen fits inside, it's only very limited in how long it can be. And I think it's like 8 or 9 amino acids long it can be, right? Because there's a groove, and so it has like a space requirement. Limitation, if you will. It's like a studio apartment. You can only do so much in there, right? Outrageous rent, but that's not really a commentary on where society is going right now. But limited size, as opposed to MHC Class 2, and we're going to see that later, it doesn't have a groove. Therefore, the antigen that can fit inside can be much longer. I think 3 or 4 times longer. So again, this is MHC. MHC1 is the first thing we drew, and then MHC II, the second one we drew. My pen isn't working all of a sudden for some reason. Anyhow, we'll figure it out. Okay, and then we talk about MHC Class 2. Again, they don't have the side groove, so the antigen that can fit inside is much longer, as we drew. But now you have to note the genetic component. It's actually going to be HLA-D, and the way I remember that a lot of languages, D, and I believe it actually comes from the Sanskrit originally, or Greek. I mean, there's that relation, right, in the European languages. I think Greek is there. Anyways, a lot of languages, D is like the first sound, right? The first consonant in like the word for 2. So I think of like MHC Class 2, right? And again, MHC Class 2 can fit the longer antigen. And so how do you make structural sense of this? Like, why is it this way? You have to keep in mind that MHC Class 2 is what the professional antigen presenting cells have. So what does it even mean to be professional antigen presenting? It must mean that you must be pretty good at what you do. And so you can imagine if it can fit larger antigens on its MHC Class 2 molecule than just MHC Class 1, it helps in being a professional, right? So that's kind of the logic I think you should maybe follow, or maybe you can use to follow, that, you know, having larger antigens being able to be projected on your surface does increase the likelihood that you can fit the larger antigen, and that's more potential epitopes or antigen binding sites that you can use to project to the T cells. And in this case, because it's MHC Class 2, it's going to be for CD4, aka helper T cells. And remember, MHC Class 1 shares its findings to CD8, aka killer T cells, right? So make sure you keep that straight. So now we talk about how is a protein loaded on to MHC Class 1. It's very important that you recognize that the mechanism is different for MHC Class 1 and MHC Class 2. So let's take it step by step, but I'm just going to give, if you're trying to make like a mental picture, the overall geographical difference, lots of better words for the two. With the MHC Class I mechanism, you really want to think rough endoplasmic reticulum. MHC Class II loading mechanism, you really want to think endosome. And when you consider where these antigens are coming from, this becomes more intuitive, or makes a little bit more sense. I'm not claiming anything is intuitive. I find this stuff quite difficult myself personally, but we try to make sense of things and then we work from there. Or at least I would like to think we do to varying extents. So let's start with MXC Class I loading, okay? Let's just say you have, I don't know, virus is inside of a cell. Works its way inside an epithelial cell, which, mind you, is a nucleated cell, but is not professional antigen presenting cell. So we're really expecting we're going to be dealing with MHC Class I here, right? So let's go with that. Virus enters the epithelial cell, and this proteasome it's a very important organelle that we have in this context, is going to degrade this virus into little peptide fragments, which are antigenic. And it's going to just basically spit out these peptide fragments everywhere. Two transporter proteins called TAP1 and TAP2 are going to tap in and they're going to join the show, and they're going to collect all these shredded up peptide fragments, which are antigenic, and it's going to take them to the rough endoplasmic reticulum, which you might recall from cell biology lectures or something. I don't have them, but if you saw something at some point, it's like the packing center, a shipment center of the cell when it wants to send the protein for extracellular use. And here is no exception, because MHC Class I is sent to the cell surface, right? So we've come full circle. And that's exactly what happens. So you have these TAP1, TAP2, they take these peptide fragments and they put them on to they take them to rough endoplasmic reticulum, where you have preformed MHC Class I, and then these MHC Class I with their newly loaded peptide fragments are shipped to the cell surface, where they're projected for CD8, aka killer T cells, to take a look at and sample what's the protein environment like inside the cell. And if something abnormal is detected, this will aid and of course the cognate antigen is one of those peptide fragments. It'll aid the T cell in becoming activated, right? Yes, good. So let's take this framework and then put it to the side to compartmentalize it somewhere else as we talk about how MHC Class II happens. Let me just move my head. It just says 2. Okay, didn't miss any critical information behind this. Let's talk about a potential conundrum. We're just trying to think about this as like a logical manner. Why won't endogenous peptides bind MHC Class II and make them no different from MHC Class I? Fair question. It's both MHC. Both of them display fragments, protein fragments. Why is it that 2 is any different, aside from the molecular structure we discussed earlier? What also has to do with the mechanism and how it's loaded. This makes perfect sense. They are distinctly categorized after all, right? So there is a specific mechanism in place to ensure that MHC Class II is saved for the 1. And this is going to be mediated initially by the invariant chain, which I have down here in red. So this invariant chain is almost like a lock or a clog on MHC Class II. It doesn't let any antigen bind to MHC Class II so casually. Rather, you have to get rid of the invariant chain if you want anything to bind to MHC Class II, right? And just free floating in the cytoplasm is not going to make the cut, because that invariant chain is quite hardy. So you have to break off the invariant chain. How do you break off invariant chain? Well, you have to kind of bombard it with endosomal contents, right? So the endosomes, phagolysosome, lysosome, things of this nature, they are filled with these proteolytic and highly active type enzymes that can destroy things, right? No exception here. MHC Class II molecule taken up into an endosome, right? An endosome is rich in all these destructive enzymes. This will actually destroy a majority of the invariant chain, except for a small piece called CLIP. And CLIP needs to be disposed of by by a protein called HLA-DM. Do not confuse yourself by trying to make a connection with the HLA from before. I'm sure everything is connected, but in the context we're speaking right now, just consider HLA-DM its own thing. Like HLA-DM like slides into the DMs here and kind of deals with this very type of saving yourself type romantic comedy going on with the MHC Class II loading, if that helps you. I don't know. Anyways, HLA-DM is actually going to get rid of CLIP, and mind you, CLIP is the remnant of the invariant chain. And now, inside the endosome, you've essentially bombarded the invariant chain to death in 2 phases, and now you have an open MHC Class II. And so inside, now antigen can go inside MHC Class II. But why is it specifically an exogenous antigen, something that was taken up from the outside? That's typically projected here. That's because what are inside endosomes? Usually things taken up from the outside, right? So it's coincidentally that, or not so coincidentally, I suppose, on a spiritual note, I don't believe in coincidences, but that's aside the point. But essentially what you have is that while we're in the endosome, what happens to be here? Oh, these antigens that are uptaken from the outside world, right? And so that's what MHC Class II is exposed to, right? And that's that. From inside the endosome, now MHC Class II, which has been bombarded open, combine the antigens that are present, which are usually exogenous in nature, and then they are sent to cell surface, right? So this is why I said again, I'm trying to make sense of it all. MHC Class I is rough endoplasmic reticulum. MHC Class II is endosomal. And again, CD8 killer T cells recognize antigen on MHC Class I. Helper T cells, aka CD4 T cells, recognize antigen projected on MHC Class II. Okay, so make sure you have these different concepts straight. Onwards. Okay, so now we have to talk about antigen presentation itself. This is T cell behind me. Okay. There are 3 types of cells who are professional antigen presenters. This is going to be your dendritic cell, your macrophage, and your activated B cell. These cells can capture antigen and they tack it onto MHC Class II and then they project it, right? If a T cell finds its cognate antigen on the corresponding MHC molecule of an antigen presenting cell, it is the good first step to activate the T cell, right? Because recall, T cells, it's very specific in how you need to activate them because, as we mentioned, I believe in our first or second module, activating a T cell is a big deal that can perform direct cell killing. It's a very active type cell family. Therefore, there's a lot of "are you sure" signals to make sure that you're really serious about turning on a T cell, because turning on T cell is no joke, right? So therefore, you need a co-stimulatory signal to serve as a check on making sure it's okay to proceed, and it's going to be mediated by B7 to CD28 binding. B7 is on the antigen presenting cell, and CD28 is going to be on the T cell. So just keep that in mind. A couple of things are needed to make sure that antigen presentation can lead to activation. So let's talk about the life of a dendritic cell, and I think we're nearing the end of this module. I had an exam this morning, so I'm quite tired, but we'll get through. So much like the resting phase macrophage, the dendritic cell has a very similar type of resting phase as well, in which it really isn't doing much aside from sampling its surroundings. And also, this type of resting phase macrophage does not have a lot of MHC Class II or B7, and mind you, these are what are needed to activate the T cells, because MHC Class II, we're talking CD4 specifically here. But there's not much of these going on, so not much of them here in this case. Therefore, you know that the resting phase dendritic cell is not necessarily adept at being a T cell activator, because what's needed isn't necessarily there that much. However, if an attack is detected, and there's different attack signal that we've discussed in the past, and I've listed them out here. And again, mind you, just like macrophages, dendritic cells also have some toll-like receptors, so they can directly know there's something wrong. But if that happens, then the dendritic cell goes from sipping mode to gulping mode, and it's going to gulp up its environment just so it can get a sample of everything going on. Just takes a huge sample, and then it's going to take it back to the lab for analysis. And by the lab, I really mean the lymph node. And so this is a 24-hour journey. The dendritic cell is going to run or swim, I guess, to the lymph node, to a lymph node, and as it's doing so, it's going to prepare itself to present its findings, and it does that by upregulating the level of Mxc Class II, which, as we established, is going to display these exogenously acquired antigens, as well as B7, which is a co-stimulatory needed for the T cell CD28. So it's getting ready to present while on the way. So, it's one time I was presenting at a conference, and I was preparing the presentation in the car on my way there. So, of course, I wasn't driving that day, so that's why I could do that. That's beside the point. So so now the dendritic cells at the lymph node, it's going to release certain cytokines to, yes, well, it gets there. It's going to release certain cytokines that are going to encourage traveling monocytes, which we established from before are freely going in the circulation, to mature into dendritic cells and take their place, right? So the dendritic cell leaves, but it's gone. So it actually hires its own replacement, which is kind of sweet if you think about it. And it's actually an important thing to note, because by default, a monocyte will mature into a macrophage once establishing itself in tissue. But if specific cytokines are released by a dendritic cell, it can actually stimulate monocyte maturation into a dendritic cell rather than a macrophage. And this makes a lot of sense when you consider that both macrophage and dendritic cell are phagocytic and are both antigen presenting cells. They're actually quite alike in several regards. So it makes sense that both of them can actually come from a monocyte, right? I thought that was interesting. So, but let's compare the two, or contrast the two, right? Both mature monocytes, both macrophage and dendritic cell mature from monocyte, and they take up residence in the tissues to become professional antigen presenters. But there is some difference, right? The dendritic cell is a big tattletale, right? The second it figures out something is wrong, whether it's like detecting tumor necrosis factor alpha, or picking something with toll-like receptors, or getting flooded with, certain red flag cytokines, the dendritic cell is going to gulp up everything and run to the lymph node, as opposed to the macrophage. Macrophage just gets aggressive and fights on the ground, right? So they're very different in that regard. Macrophage isn't going to run off to a lymph node. It's going to stay on the ground and become more aggressive, release more cytokines, and become more phagocytic, right? So yeah, these are the main things to discuss. And then if you want to read more into that last one, but you can kind of make sense of it. And so that was the dendritic cell, right? And to some extent the macrophage. But then you have the B cell, as a professional antigen presenting cell. Cell twice in the same sentence. Not a fan. I could have done better there. But keep in mind, B cell, because they're part of the adaptive immune system, it takes some time for them to kick in, as opposed to macrophage and dendritic cell. Those are innate, so those are always around, right? But let's just say once we're late into an infection and the adaptive immune system has recognized the antigen and produced like all the corresponding lymphocytes for it. But late in an infection, you have the lymphocytes kicked in, and specifically the B cells, and then they can actually join in on the fun, right? And in the context of antigen presentation, B cells can become antigen presenting cells, and they're actually more efficient than macrophage and dendritic cells because their B cells have some of the most specific and strongest affinity. And we're going to go through some of the processes, and I think we did in the last module to some extent. There's a couple processes in place, like somatic hypermutation and whatnot, that make sure that the B cell receptor binds very strongly to its cognate antigen. And there's a lot of B cell receptors on a B cell, so it's excellent stickiness, if you will, right? And then, upon doing that, it'll take it up, tack them onto MHC II, and then project them, right? As well as B7. Any antigen presenting cell will project B7. So basically, B cell is a late game antigen presenting cell, does its job much better than dendritic cell and macrophage, but of course we cannot do it without dendritic cell and macrophage because they're the ones who are running the show until adaptive immune system and its B cells kick in, right? So these are key takeaways from today. And I repeat things a lot, so I don't think it's necessary I repeat it again because I'm sure you're getting sick of my voice at this point. I think I want to close on talking about something called cross-presentation. And so I saved this for the very end because I don't want to confuse anyone more than I already have. I hope I didn't. We established that Mxc Class II displays exogenous antigens on professional antigen presenting cells. But remember, because professional antigen presenting cells are nucleated, they also have Mxc Class I. So actually, because they have both, they kind of almost cheat the rule a little bit and kind of like interplay between how they're used. So some professional antigen presenting cells will use their Mxc Class I to display exogenous antigens as well. And the benefit of cross-presentation is that basically it's projecting even more of the antigen on its surface. And that helps because this antigen presenting cell can actually engage both CD8 and CD4 T cells, right? So there's some benefit to that versatility and kind of cheating the rules a little bit. But just know, as a general rule of thumb, most of the time, aside from this exception, Mxc Class II is going to be for the exogenous antigen and Mxc Class I for the endogenous antigen, and Mxc Class I is all nucleated cells, and Mxc Class II is all the professional antigen presenting cells. But again, antigen presenting cells have both Mxc Class I and Mxc Class II, therefore they can engage in cross-presentation and kind of play with the rules a little bit. And I spoke already about the histocompatibility, and you can read more into that if you would like. And that's all for today. Next lecture is T-cell activation, and I guess the good thing is because we kind of talked about it today in some degree of depth. The next presentation is largely review, right? Maybe a couple details we tack on. Here are my sources, and I hope you all are staying healthy and well. And I'll see you next summer. Take care.